Sorry, I'm using the this, but I can project very well, but it's because we are streaming, so I need to speak in the microphone. So, welcome to our Sci Salon of October 17th. I would first like to acknowledge the sacred land on which we are standing at the University of Toronto. And as you know, as you've already heard 1,000 times, but it's very important to remember, this was the territory of the Huron-Wendat and Petun First Nations, the Seneca and the Mississauga of the Credit River. And the territory was subject to the dish with one spoon, one plume Belt Covenant. And I'm very grateful that we are here. I know that sometimes we don't feel like it's very important to remember this, but I think we are, especially as an institution that does science. So this Art Sci Salon is part 1 of 2, and it's a series that was possible thanks to SSHRC. Got a grant from SSHRC, and so I'm very grateful for them for allowing me to actually do this type of research, which is incredibly enriching. We also thank the Fields Institute, which is always hosting us. This is already, I think, year 4 of Fields Institute hosting us, and it's been beautiful. Actually, I wanted to show you something from the Fields Institute. So the Field Institute has a lot of groups and research groups and centers that are affiliated with the whole institute. And very recently, we got into a collaboration with CICAM, which is the Center for Quantitative Analysis in Mathematics. And there was a competition of math art, and we had a winner. So this is the beautiful artwork by Diana Armour, who is here in the public, actually. And I'm very happy that this has happened. So I really hope that there will be more of these competitions and these collaborations. So without further ado, I want to put the website on, but if you saw the website, there's beautiful image by Hieronymus Bosch, which I'm very happy that I could use because I couldn't wait to use it, from the Garden of Earthly Delights. That has some really strange creatures in it, and accompanied by a quote by Margaret Atwood, who wrote a trilogy, and book 1 of this trilogy is Oryx and Crake. And she has this beautiful description of these strange animals that are being brought to life in a not too distant future to please and to help human beings to get by with their lives and to provide spare parts in case their body doesn't work anymore. Now, she was talking about the future, but in some way there is no, like, it's not the future. We are talking about the present today. So one question that this series asks is, So besides GMO creatures like the pygoons or the pigoons, like she describes, there are also some all other organisms that are appearing in our lives, and some of them are coming out of climate change, some of them are coming out of labs, some of them are coming out of adaptation, and what kind of sense can we make of them? How can we deal with them? specifically, in this part of the Outside Salon, there will be I'm sure there will be a lot of discussions on human beings, because Françoise has just published a book, which I put there. And the title of today's Outside Salon is inspired by it. So if you want to have a look at that, we will circulate it. So you can browse through it. I want it back because it's my copy. So, and there's also a few copies of the description of the book, and also a review, early review that we just copied. And so if you want to have a look at that, you're very welcome to take one. So, but we have a lot of questions about gene editing, and so what is the scope of gene editing, and what is the goal? And is, as Françoise mentions in her book, she mentions actually Mario Capecchi at a certain point, I think at the beginning in her book, who 20 years ago asked the question about making human resistance to HIV. And this is something that has happened in reality last year with Lulu and Nana. But what was very interesting is that he was asking the question, so are we trying to correct something faulty? Are we trying to save ourselves from diseases, or are we trying to enhance ourselves? And this is a question worth asking. The other question that I want to bring in with this meeting tonight is, so we also have a lot of animals and organisms that we are being modifying in a lab. Are they all for the sake of human beings? Do they have other roles? What is going to happen to them when we no longer need them? And what is their future? So I want to throw them in before we start. So, but now we start. So during tonight, so I invited Françoise Baylis and Karen Maxwell, and they've both been to the Outside Salon before. Karen as a guest last year, talking about CRISPR-Cas9. And Françoise came to a workshop on CRISPR-Cas9 offered by Marta de Menges. And for those people who were here, you got to know her as well. But at this point, with the new publication of the book and with all the discussion that we are having, and with this series that I am trying to put together, I would like to hear from them more. And so I don't know who decided to go first. Who's going? Karen is going to go first. And I am not very good with introductions, so she will introduce herself. But you probably know, because some of you are her students, and some of you know her from the past Outside Salon. This is a very informal Outside Salon, so thanks for joining us. I'm going to turn this off.
Thanks, Roberta. All right, can everyone hear me? Okay, so my name is Karen Maxwell. I'm a professor here in the Department of Biochemistry in the Faculty of Medicine. And in my lab, I actually study viruses that infect bacteria. And so that's what you can see up here. So the yellow blob you see is a bacterial cell, and those are the viruses that infect and kill them. So just like us, bacteria can be infected and killed by viruses. And just like us, they also have an immune system to help protect them from being killed by these viruses. And this is the CRISPR-Cas system. And of course, CRISPR has been co-opted by scientists, and we've learned to put it in a test tube, to use it to cut DNA and to fix mutations in DNA. And we've also learned how to put this CRISPR-Cas system into any organism, or many, many different organisms, and use it to manipulate the genomes of organisms in really precise ways. So this is just to give you an idea of how quickly CRISPR-Cas genome editing and CRISPR-Cas technology has been taken up by scientists. So the word CRISPR was first used in 2002. So what you see here is the number of scientific publications against the years. And so 2019 is the very last year there. And what you can see is there's been a very rapid increase, particularly in the last few years, in the use of CRISPR-Cas editing, or CRISPR-Cas technology. So 2002, CRISPR-Cas, the term was first used in the literature. And it wasn't until 2010 that Sylvain Moineau at Laval University actually showed that the CRISPR-Cas system in bacteria actually cut DNA. And he showed that it cut the DNA of these viruses that infect bacteria. And then 2 years after that, Jennifer Doudna and Emmanuel Charpentier showed that you could actually take this CRISPR-Cas system out of the bacteria and put it in a test tube and use it to make very precise cuts in DNA. So you could target the Cas9 enzyme to cut whatever DNA sequence you wanted. And the following year, Feng Zhang actually showed, from the Broad Institute, showed that you could put this in human cells and you could use it to make gene edits in human cells. And since then, people have been using CRISPR-Cas to edit all kinds of different organisms, from bacteria to plants to animals. And as Roberta said, it's being used in people. And so I'm going to give you a little, just sort of a broad overview of all of the different, or a bunch of the different ways that CRISPR has been used to edit plants and animals. And I'm going to leave the humans to Françoise. She'll discuss those after I do. And so just a brief introduction to how actually this CRISPR-Cas system works and how it makes the edits in the DNA. So if you have a cell that has a mutation in it that you want to fix, so it has a DNA sequence that's undesirable, what you can do is you can use a DNA, a guide molecule, that should say RNA guide molecule. So you can use a guide molecule to target that specific DNA sequence. You pair that with what's known as Cas9. This is a protein that actually cuts DNA, so it acts like a pair of molecular scissors. And then you can also insert the correct DNA sequence. So those three components can be put into a cell, and you can correct mutations in the DNA or engineer entirely new sequences. And so you can sort of think of this like the search and replace function in your word processing document. So the guide RNA molecule targets, it scans the DNA to find that precise sequence. It then activates the Cas9 molecule, which cleaves the DNA sequence. So like molecular scissors, it cuts the sequence. And then the correct DNA sequence that you've also introduced can then be used to repair that cut in the DNA. And so, as I said, this has been used in all different organisms, bacteria, yeast, plants, animals. And so there's a lot of different ways that we're using CRISPR-Cas9 to make our lives better or to manipulate different organisms. So one way that it's being used very commonly is in food production. And so some of the examples I have here, companies are engineering coffee beans to not have caffeine. So it's quite labor intensive and expensive to decaffeinate coffee beans, and so they're trying to engineer them to knock out the gene that actually produces the caffeine. Wheat has been genetically modified, and rice and other crops to make them more drought resistant, so need less water, and also to make them cold resistant. And so you can improve yields of crops using CRISPR-Cas9. There's a company that's making tomatoes that actually are spicy. So they are taking the capsaicin gene from hot peppers, from chili peppers, and introducing it into the tomato. So you can make spicy tomato sauce from your tomatoes. You don't need to bother with the peppers. But one other reason that they're doing that is because it's actually really hard to grow hot peppers. They're kind of finicky. You don't get high yields. And tomatoes are really, really easy to grow. And you can get really large yields from them. And so this is potentially a much cheaper way to produce chili powder. The far left there is cassava plants. And these have been engineered to be resistant to a virus that actually causes leaf wilt. And it's sort of hard to see here, but these guys have been infected with the virus. You can see they're all kind of speckly and the leaves are dying, where the ones over on the right-hand side are still really nice and healthy. So, we're doing things that range from kind of trivial, making spicy tomatoes, to things that really could improve crop production in, all kinds of different conditions, cold resistant, drought resistant, and virus resistant. So CRISPR-Cas9 is also being used to engineer allergy-free foods. So wheat is being engineered to lack gluten. And so people that have gluten intolerance could then eat this genetically modified wheat. Eggs, there are 4 proteins in eggs that cause allergies in people. And so people are scientists and groups that are trying to knock out the genes that produce these allergens. So sort of a more fun thing, CRISPR-Cas is being used in pet breeding. So the Dalmatians, they have a genetic mutation that actually makes them very susceptible to bladder stones. And so this is uncomfortable for the animals, and it can be costly in veterinarian bills. So there have been Dalmatians that are being bred that mutation, that genetic mutation that makes them susceptible to those bladder stones is changed, and so they won't be susceptible to it. On the right-hand side are micro pigs. So these are being engineered in China. And what they've done is they've made a mutation in the growth hormone in these pigs, and so they stay tiny. And for $1,600 US, you can buy yourself a micro pig. There's another group in China actually that is starting to engineer pets. So they will clone, if you have a deceased pet, I saw for $100,000 that they will engineer a copy of your pet. So CRISPR-Cas is also being used in livestock breeding. So sheep and goats are being bred to produce more meat, and also more wool or hair for production of clothes. Beagles have been engineered, again, by a group in China to have increased muscle mass. And so they have about 50% more muscle mass than a regular beagle does. Again, introducing mutations into muscle growth genes to make them more muscular. And there is a group in Argentina that is doing similar things with horses. So they are trying to engineer horses to make them faster and stronger and able to jump higher. Okay, so these are the kinds of things that are currently being done with CRISPR-Cas9 by either academic research groups or companies. So CRISPR-Cas can also be used, is being used to eradicate pests. A great example is mosquitoes. A number of different groups, and I think Françoise will probably touch on this, are creating what are known as gene drives. And so this is a way to pass forward the mutation in a dominant manner, so you can engineer the mosquitoes to make them resistant to malaria. And there's a couple of different ways they're doing that. One is to make them resistant to the parasite that actually causes malaria. Another way that people are trying to do it is to make the mosquitoes sterile, so they can't produce offspring, or to make the females unable to lay eggs. And you know, this sounds like a great idea. I think ethically there are potentially lots of concerns with genetically engineering animals that you are going to release into the wild and have very little control over once you've released them. So other animals that people are thinking about engineering, again, to make these gene drives to knock out these animals. Apparently, feral cats in Australia are a very big problem. So there are something like 5.5 million feral cats or something in Australia, some ridiculously large number. And they have an enormous effect on the natural wildlife in Australia, that they are so predatory. And so it's been proposed that these cats be engineered, so genetically engineer some of them, release them into the wild, and they will spread that CRISPR-Cas mutation throughout the population and bring down the population of cats. Again, rats, there's an island in the Galapagos where rats are a huge problem, so they were introduced by humans coming in on boats, and they eat the eggs of the birds that nest there. And so they're wiping out whole, very sensitive colonies of birds. And so the idea, people have tried dropping pesticides down, but the rats eat the pesticides, but the lizards eat them too, and then the birds eat the lizards. And so trying to control them with pesticides has been a very tough problem. And so people are proposing, again, to use these kind of gene drives, use CRISPR engineering and release these genetically modified animals that will breed with the wild animals that are already there and cause the population of rats to collapse. And finally, sort of in the more science fiction, and this is maybe a little bit what Roberta was hoping to get here, people are using CRISPR-Cas or proposing to use it for de-extinction purposes. So on the left-hand side, that is Martha, the carrier pigeon, the last living carrier pigeon. She lived in the Cincinnati Zoo. She died in 1914. So there have been carrier pigeons once, were all over North America, huge colonies, and they became extinct over 100 years ago. So what people are proposing is to cross in the DNA with modern day pigeons and, through a process of selective breeding and using CRISPR-Cas9 to introduce these genes that were present in the carrier pigeon that are no longer present in the modern day pigeon, to be able to breed carrier pigeons. So they won't be identical to the carrier pigeons that died, but they will be very similar. And I think George Church at Harvard University has what's called the woolly mammoth project going on. And so what he wants to do is bring back the woolly mammoth. So the closest living relative to the woolly mammoth that's alive on Earth today is the Asian elephant. And the Church group has identified about 65 mutations, they think, that will make that Asian elephant become very woolly mammoth-like. And they are currently doing it. Last time I saw, they were through about 30 or 32 of those mutations. So I don't know if Françoise has an update on how close they are to it. The woolly mammoth may be a little far off. The carrier pigeon they were hoping to have done by 2022, I believe it is. So not that far in the future. And I think, my favorite idea of bringing back engineering new life would be the unicorn, of course. You take a narwhal, cross it with a horse, and you can end up, I think it could work.
Yeah, but that's what I meant. Like, I don't understand why they want to bring back older animals that are gone. How about bringing up some... This is like...
Yeah, I know.
I mean, there's no something behind it, but the unicorn is...
It's an excellent idea, I know. Or the dodo bird. My kid wants to make the dodican. It's... So, yeah, anyway, that is the end of my slide. So, we are doing a lot of different kinds of edits. This is just a small slice of what's being done. I think if you can think about, if you can conceive of it, you can potentially do it. And I think people are doing these kinds of experiments, and I think it's great to have these kind of public outreach things, because I think as a society we really need to be thinking about where we want this technology to take us and what's okay for people to be doing. So, and with that, I will let Françoise come up. Thank you. We don't want people to get distracted with that.
Well, I don't want you to get distracted, but I'll very silently.
Great. So one of the things that I decided I wanted to do today is to talk a little bit about the science, then talk to you a little bit about the history. And because I'm not a historian, I'm only going to start in 2015, because that's when the history intersects with my life. So then I can kind of feel I can speak with some authority. I then want to talk about this notion of broad societal consensus, because that's a concept that I have been putting out there and defending and taking a fair bit of criticism for. And so I'm going to try and explain to you why I think it's really important and why people are not so sympathetic to that idea. I'm going to touch on the ethical challenges, but I'm going to leave that really for our open discussion afterwards if there's something specifically that interests you, because I can then elaborate on that. And I then chose to speak about scientists and what might be their roles and responsibilities, because I thought it would be interesting for people to think about that and whether or not they think any one of those roles is reasonable and whether those roles should be restricted to scientists or not. So it's kind of an idea that builds on work done by people in political science to say, in a contemporary context, when you have people with scientific expertise and you have difficult science questions, what's the role of the scientist to contribute to those conversations in terms of public education and empowerment? And then I want to talk just a little bit about this notion of trust in science and where are we with that. So, will this work? Great, terminology. This is just to remind me so that I stay on target in terms of the various things I wanted to talk about. So one of the things that's interesting in terms of terminology, I'm going to be talking specifically about the human modifications that we'll be making to human DNA, and I'm talking specifically about modifications that are being made in what's called the germ cells. So in your body, you have two kinds of cells. You have somatic cells, which are basically all of your body cells except for your reproductive cells. And then you have your germ cells, which are your reproductive cells. And so that would be sperm and egg. It would be the precursors to sperm and egg. And it also includes, in the context of this discussion, the early human embryo. If you make changes to somatic cells, body cells, those are cells in a patient, perhaps, for example, and when that patient dies, so do those modifications. If you make a modification in the germ cells, or the early embryo, those modifications are passed on to the next generation, which is why we think about them as heritable. Now, one of the reasons I have this slide up here about terminology is because in the very early days, so now I'm talking 2012, maybe, in the very early days, we used the term germline genome editing to refer to all kinds of manipulations being done in eggs, in sperm, early embryos, etc. But there was some slippage happening because some people were talking about doing that work in the lab, and some people were talking about doing that work in the lab and taking it into the human in order to have people give birth to new humans with genetic modifications. And so one of the things that I've done in terms of my own work, because I want to be able to focus on the ethics, and people might have different ethical opinions about work that happens in the lab with these materials versus work that happens in the lab and then gets transferred to a human for reproduction. So in that context, I use the term germline genome editing to strictly refer to manipulations in a lab, and I use heritable genome editing or genome modification for when you are taking those modified tissues and using them for a reproductive project. Okay? So the reason I'm insisting on that a little bit is because I'm trying to be really consistent in my own use, but it's for you to understand in the context of discussion and debate, people will sometimes use just the one term, germline genome editing, but they may mean one of those two things. Okay. So the next thing I want to do is talk to you about recent history, and as I said, I'm only going back to 2015. This is Dr. Huang. Dr. Huang is a scientist in China who, in April of 2015, published a paper in which he shared that he had done work in what are called non-viable human embryos. So these are embryos that cannot be transferred to a woman in the hope of expecting a pregnancy because they have some inherent defect. And we just use this term generically. In this particular case, these were triploid embryos. One of the things that becomes important for you to understand is he does this work in the lab. They're non-viable embryos. He destroys them. He does not transfer them to a woman in the hope of establishing a pregnancy. And he identifies, amongst many other things, that the success of the work that he was doing was not very high and that we really needed to think about the ethical issues. And in response, there was a great hullabaloo. Many people objected. They said, this is terrible. You shouldn't be doing this work in human embryos. It's not as if people didn't know you could do it. It's just nobody had done it before, or if they had, they certainly hadn't publicized it. They hadn't published it. He does this work. He publishes it in April And as a result of that, there is the first international summit on human genome editing. That's a reference here, a global discussion. It happens in Washington. It happens in December of 2015. And in terms of full disclosure, I was on the organizing committee for that meeting. And at the end of that meeting, we issued a statement. And this is just an excerpt of the statement. You can find it on the web if you're interested. I don't want you to think I'm taking it out of context. But speaking here, and we actually used the words germline editing, but this is an example of where what we meant was heritable genome editing. And what we said is that if you're going to do this work, you have to meet these standards: safety and efficacy and broad societal consensus. Now, I will tell you, as somebody who works in philosophy and in ethics, this is a two-part ethics framework. It's got to be the simplest ethics framework that anybody has ever put out there and defended, because ethics frameworks are usually long and complicated, etc. Now, I think it's lovely and simple, and at the same time it's exquisitely complex. Why? Because nobody actually knows what this means, right? So here you have this very simple ethical framework. It says two things: safety and efficacy, broad societal consensus. And at the end of the day, I'm saying we don't know what it means. Why? We want safety because nobody wants to hurt anybody. We want efficacy because you don't want to sell people a bill of goods. You want to know that it works. You want broad societal consensus because the areas into which you're going are potentially controversial, and what are the kinds of changes you might want to make to the next generation of humans, and who gets to decide what those changes look like? Is it for one scientist to decide on his or her own in their lab? Well, I think this is going to be a cool modification, so I'm just going to make that. And all I need to do is find some woman that wants to accept the transfer of my genetically modified embryos, and we're off to the races. And so what was being said there is, no, we need to know something about safety and efficacy. And the reason I'm saying we don't know what that means is because it is never going to be 100% safe. It is never going to be known to be 100% effective. So when you do this research and you move into the lab, somebody is going to have made a completely subjective decision that we're good enough to go. It's safe enough. We think it's effective enough. But that's a value judgment. And if you think about things that we do now, right? If we're treating cancers, we have interventions where we go in and do a lot of work thinking, hoping, if we're lucky, we're going to have a 20% chance of success. But in some cases, we think 20% is good enough. In other contexts, we might say that's not good enough. Go back to the drawing board. You know, you've got to get your anticipated success rate much higher than that before you can go out to patients and say, this is worth trying. So it's to say we don't yet even know what safety and efficacy is, even if we think about it as somehow objective. It's not. It's a subjective concept. Broad societal consensus, well, my goodness, consensus, what does that mean? And who do we think is broad society? And who are we going to get to agree, right? What happens next is in 2018, this is Zhangqiu He, another Chinese scientist, and he does different work in a lab. He is actually working with human embryos. He's looking to modify what's called the CCR5 gene, and what he hopes to do is to provide the next generation with resistance to HIV. So that's the modification he wants to make. He does his work in the lab. He transfers embryos to a woman, and 2 girls are born, referred to by their pseudonyms Lulu and Nana, who are supposed to be resistant to HIV. Now, when he does his work, the second international summit that takes place in Hong Kong is not a response to his work, as was the first case in 2015. This meeting was already scheduled to happen in Hong Kong. He announces this indirectly in response to a leak by MIT Technology Review, and he tells the world, I've done this. And you can imagine he certainly gets a lot of attention, but he in effect hijacks an international meeting because now this is all anybody can talk about. Oh my goodness, we have created the first genome-edited babies. There's all kinds of debate and discussion about how good was the science. Are these children going to have problems? You know, who knew? Why was this happening in secret? So there's a lot of ethical controversy. I was not on the organizing committee of this particular meeting, but their organizing committee does the same thing, which is at the end of the meeting they issue a statement. Now you'll recall the statement out of 2015 says safety and efficacy broad societal consensus. And that's in response to a scientist working in non-viable embryos in a lab and discarding them and doing all of this consistent with what would have been the norms in his country and, in fact, international norms, but for the fact that nobody had done this particular technique in an embryo. In this case, we have genome-edited babies, and this time the conclusion is it's time to define a rigorous, responsible translational pathway forward. So I want you to think, well, what happened in 3 years that we go from being super upset and angry with some scientists for doing work in a dish, and now we're championing, even though we're telling the world that this particular scientist was irresponsible, that there wasn't safety and efficacy, and that is one of the criticisms, there wasn't safety and efficacy, but the whole idea of broad societal consensus has dropped off the table because in the interim, in those intervening 3 years, people are thinking, that was a mistake. We should never have said broad societal consensus because we're never going to get it. So actually it means we'll never get to do this science. Okay? And so what they say now, we've had babies made. What we need is a responsible pathway forward. So this happens in March of this year, 2019. And again, in the spirit of full disclosure, I'm an author on this paper. First author is Eric Lander, and what I want you to know about this particular paper, which comes out in March of 2019, it's largely in response to the decision of the committee in 2018. We're saying we need time to think this through. The only way to get time to think this through is to have a moratorium. And during that moratorium, which we suggest could last for 5 years, this is the work we need to do in terms of ethics and governance, which would be about finding a way to think, should we be doing this? And if so, what's the direction in which we should? Now, the reason I want to mention this is because what this paper does is two things that I think are important. It makes explicit the fact that there are two different camps amongst the scientific community. So I'm not a scientist, even though I'm a co-author on this particular paper. The reason I say that, it makes explicit that there are two different camps, is that half of the people who were part of the 2015 statement sign on to this, and the other half who were part of the 2015 statement do not sign on, and moreover, they are the authors of the 2018 statement. So if you actually look at the names and the authorship that's being attached to it, those who signed on in 2015, by and large, are co-authors of this paper. Those who were part of the 2015 summit and the 2018 summit are now standing behind the 2018 summit's conclusion. The other thing that's particularly important is you heard before that it's 2012 that Jennifer Doudna and Emmanuel Charpentier do their work in the lab to show that CRISPR-Cas9 can make these cuts in eukaryotic cells, and it's a year later that Feng Zheng does the same work but in the human model in 2013. These three scientists are widely recognized as the CRISPR pioneers. Two of those three scientists sign on to this. One of them does not. So Jennifer Doudna does not, and that is in response to more than one request to ask her to please sign on. So it's a purposeful choice on her part as a scientist to say, I don't support the idea of a moratorium. Now, the thing I want you to hold on to as an idea in terms of, a public perspective is, what's a moratorium? A moratorium, by definition, is a temporary prohibition. In this paper, we make clear that it's temporary. We say 5 years, and we actually outline the kind of work that has to happen within 5 years such that you could then, move forward in whatever direction or work has gone forward. What's interesting is, at least in the academic circles, it becomes a debate about the M word, and they literally use that kind of language. We can't use the M word. And the fascinating thing to me is there isn't a single scientist I know in the United States and most of Europe, and I don't know in certain countries like, say, Russia or China, etc., that wouldn't say it's premature to go forward, that wouldn't say it's irresponsible to go forward. So they all believe that. So ultimately, they all believe that you shouldn't do anything for the next little while, however the next little while is. But it's interesting that this has become a flashpoint of debate, that we can't have a moratorium. This is Denis Robakov. He is a Russian scientist. He has not yet done the kind of work he's proposing to do, but what's important is that in June, after this paper has been published, he says, I too am going to do some genome editing in humans in the context of making heritable changes. I too am going to make changes to the CCR5 gene, so I'm looking at resistance to HIV, but I'm going to do a better job than Jian He. I'm not going to do this work in the male, where we could have addressed the concern by doing something like sperm washing. I'm actually going to target females, where what we're worried about is vertical transmission, that the pregnant woman would pass on the virus to their offspring. He gets a fair bit of pushback in terms of, how are you going to find these women? What's going to be the population? Why do you think it's going to be any better? That even if you were successful, you're really only looking at a subset of ways or forms of HIV that could be transmitted. It's not going to be all HIV, etc. And in the context of this pushback, he says, okay, if I can't do HIV, I can do dwarfism, blindness, or deafness. Okay, those are his 3 examples. And then people are saying, like, what? And this is in a context where you're trying to say this is potentially important work that could be offering therapeutic interventions for people with serious underlying, perhaps fatal illnesses in the immediate postnatal period, and you come up with these 3 examples. And since then, he claims to have found a couple where there is heritable deafness, and the gene that he's interested in is referred to as the GJB2 gene, which is known to cause a particular kind of deafness, and he says, I'm going to go forward. I have looked very carefully at the legislation, the research guidelines, etc., and there is nothing in the legislation in Russia that would stop him from doing this. There is one clause in the rules around assisted human reproduction which would require for him to get a kind of ministerial or governmental approval, and that leaves it open to what does the government think is or is not appropriate. So one of the things that I want people to think about, and the reason I think this is important, is I believe that most scientists who are working in this area have very specific kinds of goals and objectives. So I'm only talking about the positive goals and objectives, and some of them are what I would describe as other-oriented. I want to make the world a better place. I want to help patients with certain kinds of conditions, etc. And some of them might be self-interested. I want to win a Nobel Prize. But there's not a lot of scientists who don't want the approval of their peers, who don't want to be celebrated for whatever work it is that they're doing, and they certainly don't want to be shunned by the scientific community. So imagine if you had a really clear message that says, we have a moratorium. I think most scientists would want to respect that and to be seen to be working with the scientific community to try to figure out, okay, what are the responsible rules and regulations and contexts within which we might do this work? But we don't have that because we have this divide amongst members of the scientific community about what should or shouldn't be done. Now, in this case, I put the World Health Organization because there isn't a 3rd international summit yet. There might well be. But there is now this committee which is tasked specifically to look at issues of governance and ethics. And in that context, the Director General in the summer of 2019, in response to what Dr. Robakov said he was going to do, issued a statement saying or calling upon countries, please, can you do what you need to do locally within your nations where you have authority, responsibility to ensure that we don't go forward until we've had a chance to work on this. He too is not going to use the word moratorium or anything else, but you can see that it's an effort to say to independent nation states, because nobody can tell an independent country right now what they can and cannot do, but all you can try to have is moral suasion and a certain kind of communal understanding about what would be acceptable. So let me just share with you now this next thing. What is this broad societal consensus that I and others advocated in 2015, continue to advocate for now, and why do people resist that idea? Okay? So broad societal consensus literally gets used for the first time in that locution in this document. Other people have talked about broad societal consent, and you can go back and find some early works by David Suzuki where he uses that language. But when it moves to this notion of broad societal consensus, the only reference I can find to it is in this document, in this gene editing document. So I've been spending a lot of time since then trying to unpack this concept and, quite frankly, proselytize a little bit around what is this idea and why should people be enthusiastic about it. This is a whole talk that I actually give, which I've collapsed into a slide. And what I try to say here is the decisions that need to be made around heritable germline genome editing are too important to leave to the scientific community. Okay, so that's what I say. They're too important to leave to the scientific community. So part of what I'm saying there is the discussions to happen can't happen in the lab. I then think, okay, well, if they're not going to happen in the lab, where should they be happening? And some people think that they can, they should, they will happen in one or two other environments. One of those would be in governments, right? Like governments will decide. They'll set policies, rules, regulations, guidelines. And other people think about it as the boardroom of companies. And that's where these kinds of decisions should be made. And I say, that's not good enough, because that's still a very particular kind of elite group of people who would be making those decisions. And both of those environments are doors that are closed. They're not open to the general public. And I argue, if you think about the human genome, what you should be thinking about is, it's kind of like the only thing that we all share. And in that context, I think about it as a metaphor for something that we have in common and that we ought to be thinking about. If we share this, then if we're going to start making changes, if we're going to take over, in some sense, the evolutionary project, we should all have a say. And so what I try to say is that these conversations need to happen in the kitchen. These other people have to go in the kitchen too, right? Like they have to eat. Scientists eat, people in industry eat. So do people, in government, etc. But everybody does. And what I try to get people to think about is just think about what are the kinds of conversations you have in kitchens. And even the conversations in your kitchen, I am sure, are different than the conversations in the bathroom, in the bedroom, in any other room in the house. There's a certain kind of relaxed, jovial conversation that happens in kitchens. It's collegial, you're sharing food, etc. And I think what I'm trying to say is that these become sort of friendly conversations typically. What would it be like to have a friendly conversation about what the future could be? And then I ask the question, and what if you imagine going outside? Because often there are doors outside of kitchens. And I think, what if we were having a picnic? What would it be like to think that that's where the conversation should happen, and those are the people who get to participate in the conversation? So that's the first thing that I'm talking about when I say broad societal. I'm saying society is more than the people who currently have power. Now, that doesn't mean that they are not experts that have very important contributions to make, but I'm actually making a claim for other kinds of expertise, right? Lived experience and other kinds of priority setting. And I think that that's an important thing for us to consider as well. And then what I do is I actually show these are not all novel ideas. And I actually go back to interesting work done in the mid-1980s, and this was by women who came forward who were fighting for peace. I probably shouldn't say fighting, arguing, hoping for, advocating for peace around the deployment of the Pershing missiles. And because these were women coming from across the United States, they had to find ways to come to agreement on what they were going to say publicly. And they actually came up with 5 principles for conversation. And I have bookended them by adding inclusivity and benevolence. And I'm not going to go through this here now, but we can come back to it in the discussion period. But largely it's about rules for conversation. And the most important thing to understand here is that ultimately consensus is not about unanimity. We are 7.7 billion people on this planet. We are not going to agree on anything, really. It's about unity. Unity is about a coming together in terms of a situation whereby we think these are the rules under which we can govern ourselves, and these are the things that we're prepared to agree to in order for us to be able to carry on. And there are many ways in which we can do this, and the very simplest example that I often give to people is we've done that with embryo research. Lots of people don't think we should be doing embryo research. Embryo research is happening. We have an understanding that you don't go beyond 14 days, and there's a lot of reason and history behind that, but it is not the case that every single country has a law that says you can't go beyond 14 days. So it becomes an example of, look, we have got consensus on something. What did we do there? How did we generate a consensus, an understanding, and people are holding themselves accountable to that? Ethical challenges, I said at the beginning, I'm really only just going to name them for you so that you understand how complex the issues are. And by and large, there are benefits to this technology which are significant, and they fall, I would say, largely into these 4 groups. Okay, so we can imagine prospective parents would benefit, future children would benefit, society would benefit. You can even make claims that the gene pool could benefit in terms of, so long as you keep enough diversity, you remove certain kinds of defective genes from the gene pool. But there are also potential harms, and here all I want to point out to you is that the harms devolve onto the very same groups, but I've made a point of pulling out and highlighting the women egg providers and the women who may serve as gestational carriers. Because when we talk about this technology, honestly, a lot of the circles that I move in, they talk about this technology as though, yeah, we just got all this tissue in the freezer, let's go get some of whatever we need and let's play around with it in the lab. And I'm trying to say, where do those tissues come from? They actually don't come from freezers, right? Ultimately, they started off in women's bodies. And it's very easy, comparatively speaking, to get sperm. It's very hard to get eggs. And it has potential harms for the women who do this. And we don't talk about that. And so that's one of the things I try to bring to the fore in terms of thinking about what are the implications of just barreling along and doing this science. These are just some of the broad categories that I discuss in terms of what might be some of the harms and some of the wrongs. And the point that I make as a category issue is, what does it mean to talk about wrongs? Well, the example I give is I know lots of ways to safely and effectively destroy the Mona Lisa. And most of you would say, well, yeah, well, that's wrong. You shouldn't do that. I can think of many ways to safely and effectively destroy old-growth forests. And many people would say that that was wrong. So part of what I'm exploring here is why safety and efficacy wasn't a sufficient standard in 2015, and why we added broad societal consensus, because we need to come together to figure out when is it going to be wrong, even if it's safe and effective. So it is possible that we could reach a point scientifically where we say these interventions are safe and effective, but in certain conditions we might still say, yeah, but that's wrong, we don't want to do that. And in other conditions we might say, yeah, it's safe and effective and it's right, we do want to do that. I'm going to move on to what is the last part here, which is the scientists and roles and responsibilities, because I just thought it might be interesting for you to reflect on what does happen when you have people that have expertise that you, as interested, civic-minded citizen who wants to participate in broad societal consensus is not going to have that knowledge, and you're going to be dependent on them. So I have views about climate change, but I don't understand the science. I have to defer to my favorite scientist, right? And I have to figure out who's my favorite scientist. Who do I want to defer to, right? When I'm trying to understand a complex debate. And part of what I'm trying to say here is that at the end of the day, I'm going to try to find somebody whose values align with my values, and then I'm going to trust that their science supports those values. I could be making a mistake, but that's often what happens. So this is not my work, even though I've taken it and applied it in the context of the book, and I have a whole chapter on this. It's work done by an American political scientist by the name of Roger Pilkey, and he talks about idealized roles for scientists, and these are my terms, but they're really building on his work. I talk about the pure scientist, the science analyst, the issue advocate, and the science diplomat. Very briefly, who is the pure scientist? The pure scientist is the person who says knowledge for knowledge's sake. That's what I do. I'm just out there generating truths in the world. It's not my responsibility how people use the knowledge that I generate. Some people will use it for good. Some people will use it for evil. I'm just interested in truths. And I'm going out there into the world and looking for truths. And in the context of this work, Francisco Mojica has self-characterized himself that way. He says, I'm just interested in answering questions. And he is the first person to have coined the term CRISPR and applied it in this context. There are other people I could put there, but I put him there because he self-identifies in that way. That's what I do. I'm just interested in interesting, cool questions and finding answers to them. You then have the science analyst, and the science analyst, like the pure scientist, wants to generate new information, but is deeply committed to trying to help others, and specifically the public and policymakers, to understand and to interpret that information. So it's not just putting out information there and hoping people will get it, but actually helping them to be able to discern what's the relevance or the importance of what is being shared. And I think what's important here is this idea that science has a dual purpose. Yes, it's about discovering and disseminating truth, but it's also about promoting social reform, and so that's why you help with the interpretation relative to your goals in terms of social reform. I've put Francis Collins, the director of the NIH, in this category, though I will say to you up front, he is not always in this category, which is what allows me to make the point that these are idealized roles, and it's not the case that, a scientist fits into one box for their life, for a time, for an issue. People move around. These are, in some sense, caricatures, but they're meant to help you understand how people contribute to debate. The next one is the issue advocate. Like the people before, generates information, assists with interpretation, but then actively engages in supporting policy options because they actually have a view that there's a particular right way to understand the science and how the science will contribute to making a better world, and they emphasize that. So they're basically aligning themselves with a certain set of political goals. So again, if I take climate change as an example, there are people who will tell you, this is good science, this is bad science, and it's in support of a particular view. If we think about the use of cigarettes, it does or it doesn't cause cancer. People are aligning their skills and their talents with a particular view. This is Josiah Zahner. He's a biohacker, and he basically says, look, procreating by having sex makes a lot of mistakes, and if this is going to help fix those mistakes, I'm all for it. And he also is, has a very particular view about democratizing science, etc. And he's out there advocating strongly for a viewpoint. He doesn't have any sense of himself as the pure scientist in his lab just generating knowledge. He's out there saying, these are the things I believe in, and I'm using my science to help you believe and support what I believe in. The last one that I've put up there is the science diplomat. And what I think is important here is I think the science diplomat is trying to promote collective decision making and is trying to help people align the science with values. And the thing that becomes, I think, important about the science diplomat is not that they are not also issue advocates, but I think they get to their advocacy after they've done sort of their collective decision-making work. Because if you do it the other way around, you're already sort of polarized, you're already positioned in the debate. So it's not that they don't have views in terms of what ought to happen, but they really are trying to sort of say, how can I bring people together? How can we find out what we have in common? How can we use what we know to move things forward? I'm going to end just with this debate about trust in science, and what I wanted to show you here is this is the level of trust worldwide. It's relatively recent data, June of 2019, and if we just look at the United States and Canada, high trust, 26%. That's actually not very high in the grand scheme of things. You would want that to be at least 50% of your population trusts your scientists, right? I mean, for me, this was really quite shocking to see that it was that low. Now, you can choose to see it positively and say, yeah, but, if you add the 50%, that's sort of medium trust. But that kind of doesn't seem right for me. Not if you're going to be handing over in some way, shape, or form decision-making about all kinds of really important things. So I want people to think about that, and I'm going to, in the interest of time, skip through this because I just want to show you a very short video, and then I want us to have a conversation. The reason I'm going to show you the video is because I think it's what contributes to undermining trust. This video is going to start with this image here, which is the film, but I want you to be able to read the tweet, and maybe you can't from there, so I will read it for you. The National Academy of Sciences, about 10 days ago, put out a tweet. The tweet says, dream of being stronger, and then it's got one of those emojis of like a good muscular person, or smarter, and then it's got a little picture of a brain. Do you dream of having a top student or star athlete or child free of inheritable diseases? And then we've got your athlete lifting weights, doing things. Can human gene editing eventually make this and more possible? Hashtag the science behind it. So this is the National Academies of Science in the United States, and they are tweeting about it. They are sending people to their website. And I just want to show you this film. You're going to help me turn the sound on? Okay, let's see if we can get the sound, because it really makes a difference, and I think it's really important in terms of this issue of trust in science.
Do you have
well, I think this was it.
No body hair, really?
It was working before. Just give us 1 minute.
I definitely want to require less sleep.
Have a small stomach.
Get rid of my buffalo hump.
Create the perfect human being.
No, come on. No, come on.
That's pretty cool.
If it would be possible to pay and have the physicality we want, would we do it?
I don't know how I feel about that.
I mean, you want the best qualities to be put into your offspring. I want my child to be the best version of not just me, but him or her. So if I can control that or have some control over that, I don't mind it. I don't hate it.
I know.
I'm chomping at the bit because as the only actual parent here, because I have given birth to 3 boys and they're all different, I wouldn't change anything about them.
I totally get what you're saying, but I'm definitely sure that I would be a hypocrite if I say that I wouldn't change it, because if I had the chance to choose the best DNA for my child, I would definitely want her to be smart. I wouldn't like her to be an introverted child. You know, I would take that from there.
But what if the world was full of extroverts only?
That's
oh my
the thing.
gosh,
That's
that's
the
a
thing.
problem.
That's the thing. But there's a thing. All parents wouldn't choose the same, because all parents don't have the same tastes. So people would always be different. They wouldn't all be the same.
For my dad's side of the family, they all have like diabetes and high blood pressure, and my grandfather passed away from a heart attack. So I would take that out for sure. I don't want my kids to deal with it.
Yeah, with diseases,
you know.
I definitely get you. But for example, I'm ADHD. In the world, it's considered a disability, but I don't consider it that. It helped me when I was volunteering at juvenile detention. And a lot of kids that end up in juvie are ADHD or bipolar or depressed or whatever it is. I go, I understand you. I know where you're coming from.
My answer, if we had to mess with embryos, if we could prevent like all these diseases that we've been talking about, all the high blood pressure, heart attacks that are happening, if we could do that, I guess I would say yeah, even though I don't agree with changing someone's hair color or how tall they are, because I don't care about that. I just care about their health. Yeah. So I guess I would say yes.
If that could happen, it had to be a thing for everyone.
Gotcha.
Like equal people, rich, poor people, everyone needs to have access to that.
Mm-hmm.
But I know this would be really hard.
It's impossible.
So probably no, I would say no.
I don't feel either anyway. I just know that I wish I wasn't dyslexic.
But I think you need to celebrate the dyslexia and figure out how you are different and unique by that and how special you are.
Usually, I don't like to tell people that I'm dyslexic because they immediately feel sorry for me and I feel like something changes in the way they perceive me.
There's a lot of people that are dyslexic and have overcome that. It's not a disability, sweetie. It's actually something we need to celebrate.
It's very nice to hear. Thank you.
See, your legs are fine. Your legs are great.
It's nothing.
Legs are great.
These chicken legs are gone.
I was gonna say.
No, I don't want to do that. I just want to sort of end by saying that's the National Academy of Science, and that's supposed to be the science behind it. What science did you learn? And one of the things that's deeply problematic is you're talking about things that are kind of crazy, like, you know, in terms of what might be possible. You actually had only one person even talking about, you know, illnesses or diseases. And he wasn't talking about single gene diseases, which you might have a chance of looking at. There's no attention to gene-environment interaction. There's nothing, but this is the science behind it. This is the National Academy of Sciences. And the first image is like, you know, what are you going to fix? And the reason they're laughing at the man at the end is because he said, I have these chicken legs. I want to get rid of my chicken legs. And the woman who has the buffalo hump. And, you know, you're just sitting there, how could you possibly have thought that this fits under the banner of public education? Well, within about 24 hours, myself and a number of other people raised a lot of concerns. The tweet was erased, the video has been taken down, and it's not available anymore. If you were to try and find it, you would get to this page. It says, note to readers, this page on the topic of human genome editing has been deleted. We are concerned that the content, including a video, left the impression that the use of genome editing for the enhancement of human traits is permissible or taken lightly. Were you left with that impression? No. So one of the things I want you to think about is I really believe that these are important questions. I do genuinely believe we are on the cusp of taking over the human evolutionary story. I think we can do that with a fair bit of hubris, Or we can do that with a fair bit of humility. We can allow certain people to just kind of march off into the sunset and do whatever they think is right for the next generation, even if they really are genuinely motivated by all the right things. Or we can say the decision belongs to all of us. And what's interesting is around this language of broad societal consensus, people are uncomfortable because they think consensus means unanimity. They don't understand the concept of unity. And what that means is they don't understand that even if we never achieve anything close to consensus, if we have that as a goal, we will have different kinds of interactions with each other. We will have different kinds of conversations. We will at least try to understand the different point of view in order to try to bring us closer together in terms of understanding what's important and what isn't. What's interesting is that the people who don't like the term broad societal consensus have found themselves using another term. So Victor Zhao, who is in charge of the National Academy of, I think it's medicine as opposed to science, one of the national academies, I don't want to get it wrong, has actually said, no, we need broad scientific consensus. And I thought, really? Okay, you've laid your cards open. In the United Kingdom, they say, no, we need broad societal debate. And I say back, okay, and when's the debate over? Like, what are the rules for calling a close on the debate if consensus isn't the goal or objective? So what I find interesting is that in all the pushback against broad societal consensus, you're actually having another kind of debate ultimately surface. So, in closing, my book is a call to action. It really is aimed at a general interested public, and it's about empowering people. It's about giving them tools, both in terms of science literacy and ethics literacy, so they can participate in the conversation. So thank you very much. I appreciate your attention, and maybe we can have a bit of a conversation now.
Okay. I have a list of stuff. Okay, so how about, do you want me to take a seat myself?
Sure.
Okay, so let's start. I'm going to just put this a little bit closer. Oh yeah, no, like I'm just going to keep the brakes. I don't want the... I'll keep it. we just move that like a corner of the road. Chair.
I think we need to... We have one here for you.
Oh. So in the middle? Oh my God.
I think we have to share this microphone.
Oh, yeah. Okay, so I know that you guys have a lot of questions. I have a lot of questions too. So, but because I'm the host, I will let you have questions and then I'll. Otherwise, I can start. I guess for the first question. I mean, one of the things that I was thinking about when I heard both of you speaking was, so I kept asking myself, so why do we try to bring back the passenger pigeon? Why are we trying to? No, why are all these scientists trying to create these people who are resistant to HIV? And I know there's been a lot of research to eliminate Huntington's disease. So why not that? And why is the HIV thing so much more publicized? So all of these why that I'm asking myself. So I'm trying to grapple with it. So are we making these selections? Are we? So I'm going back to this idea of the announcement versus the correction, or who does this benefit? So,
Start with the passenger pigeon.
The passenger pigeon, the unicorn.
I think in some cases, people are choosing to do it for profit. I think certainly with crops that there is an aspect of profit. I think in the case of the passenger pigeon and the woolly mammoth, there is an element of, I want to see if I can do it. I mean, I think it seems particularly with the woolly mammoth, well, I don't know, and the passenger pigeon, I don't think we're going to start using them again to send letters around.
Do you think that this is easier? So this is easier to do?
as opposed to other achievements, or like as a scientist, do you think that this is a scientific endeavor that can be achieved?
Like those two particular ones, as opposed to something else? I mean, it could be.
I don't know, like I'm not a scientist, so I was kind of trying to understand.
Yeah, I don't know. I'm not sure why the passenger pigeon. It wasn't clear to me. Maybe, I don't know. I don't think that birds are easier to manipulate than other animals.
I think that in the human case, most people who are arguing for this are not starting at the extreme with the enhancement ideas, which is why the tweet and the video from the National Academy of Sciences was seen to be deeply problematic, because most of the scientists who are looking at work in the human condition are targeting things that might broadly be understood under the banner of genetic illness or disease. But it's also because of that that some people were quite concerned about Denis Robakov saying that he was going to make changes to a gene that is associated with heritable deafness, because that's a situation whereby the deaf community has been quite vocal that they don't see themselves as having genetic disease or illness, that they're a community, that they function differently in the world, but they have everything that everybody else has, including a language, right? And that this, for many people, epitomizes one of the problems. Who gets to decide what is an illness, what needs to be corrected or changed? And I think that's a particularly interesting point. Having said that, let me explain what I understand the science enthusiasts around this to be saying. They point to patients with very difficult life-threatening conditions. One example is, in fact, Huntington's disease, and I discuss this example in the first chapter of the book. And I explain that if you have, children with Huntington's, you yourself, as a person with Huntington's, will experience a number of debilitating symptoms on the path to an early death. And it's very difficult to then learn, because it's a late onset disease, to learn that you have this condition and there's a very real chance you've passed it on to your children, and then perhaps to learn that your children have this illness. So in that context, people understand why it would be a good thing to try to relieve that suffering, to try to be able to offer some kind of a cure, and that you would have that as a goal or objective. So then the issue comes along to say, well, if we're going to try and treat this patient, why would we treat this patient, allow them to go on and reproduce, and then have to treat their children? And then those children will grow up and they'll reproduce and they'll have children with the illness, and we'll have to keep doing this in perpetuity. And so they say, look, one and done, let's fix it. Let's fix the somatic cells, let's fix the reproductive cells, let's just, eradicate this disease, because that would be an example where they don't think there would be a lot of controversy about whether or not this is something to keep within the human experience. But in that context, there are a lot of facts that have to come to bear, which is, how do you know that you have Huntington's disease unless you have a family history? And you might not know that, because people may be carriers but not have experienced the illness. And there's a number of other things I could say, but the most important thing in terms of the debate right now is people say back. There's a huge difference between the alternatives which are available and families could use, and the alternative you're trying to develop. What do I mean by that? Currently, if you have a particular genetic illness, we are able to offer you many other ways of having children. You could, for example, adopt. You could, for example, use assisted reproductive technologies with donor gametes or donor embryos. You could use your own gametes and embryos, understanding that a certain percentage of your embryos may be affected with the condition that you're trying to avoid, but then you would do pre-implantation genetic testing, you would identify the healthy embryos, and you would only transfer the healthy embryos. So people say there are all kinds of other ways that you could go about having children that would be genetically related or not, depending on the technology used, and would not have the heritable illness. And they go so far as to say those options would be better because you're not embarking on a design project. And the problem, people would say, with embarking on a design project is you're ultimately commodifying children, and you're engaging in something that is one step along the path to things that ought not to be done. So some people will bring forward slippery slope arguments and a number of other things. So that's a sort of a short version of why some people think, of course, we should do this, and other people think it's deeply problematic.
So I'd just like to add a comment to that. You brought up Josiah Zahner. Right. And so Josiah Zahner, I was, about a year ago, I was in a debate with Josiah Zahner. And so he's a biohacker. He thinks everyone should have free access to CRISPR technology. He sells DIY kits from his garage. And he thinks that anyone should be able to make any edit that they want. And he actually said during the debate, what's wrong with having a world full of tall, thin, beautiful, blonde people? And I don't know. I think that's something that's open for debate. But there are people in the community who think this way, and who, I think some of it is shock talk, maybe. But, how far do we take it once it becomes safe? And I think there will be a point in the probably not too distant future where it is safe. And I think things right now we can say, well, it's ethically we should be treating people with these horrible diseases. But how far do we take it? What if, you want to have a tall child or...
There are people, right? Okay. 1 and 2.
Thank you. And here the three of you, or thinking. But what I gather, and I may be wrong, is whether there are bits that can be cloned in perpetuity, or kids that you may want to have another chicken leg. It's a very emotional component. And when it comes to trust in science and the debate that we all should have around this issue, the trust in science is slim to none, depending on where you are, but it's below a third. Doesn't matter where you are. So, how do we educate people? How do we get them to understand? And how can we be sure that we do not necessarily carry on our own life in this teaching?
I think I'll just say one thing, which is I actually am of the belief that we can't avoid our biases, and therefore what we have is an obligation to make them plain. So, in other words, if I'm going to advocate for a particular viewpoint and I'm a scientist, I don't just tell you, I'm a scientist, listen to me, I have authority, I have knowledge, this is what you should do. But you say, I'm a scientist, I have a lot of knowledge, etc., that I can bring to bear, and I have these biases. We all do. There is nobody in this room that doesn't have biases. And I think part of that is you just make it plain and transparent. So if we share biases, then maybe you want to defer to my expertise and knowledge. But if we don't share those biases, maybe you'll be less inclined to have trust in what I'm going to say or not. But I think that's in part what the science diplomat is helping people to do, is helping them to align values with science. And don't misunderstand me, I'm not arguing for a fact-value distinction. I don't think that there are objective facts in the world. All of that is filtered through our lenses. I mean, to just see a question as important is already to bring values to that question, to have determined that it was an important question. Mm-hmm.
I think it's important for us to say, as far as I know, to the best of my knowledge, and so on and so forth. So there's no blanket.
Oh, I met a lot of different scientists. I'll let you comment.
Because they may hear that it's great to be able to do a lot of things with these tools, but how do you teach them about the consequences? How do we educate? Okay, how do we educate people so they know what they're debating in their kitchen or at their picnic?
Yeah, I think that's a really tough question. And I think, as a scientist, I am spending my time in the lab and I'm with my students who have the same expertise as I do. And with other scientists, we speak a particular language that isn't widely accessible to a lot of other people. And I think that, people are becoming more aware of how important science communication is. Universities are starting to pay a lot more attention to it. We're trying to train our students to be, to help create scientific literacy in the public. I think these kinds of outreach programs organized where more of the general public come in is very important. And, I think, I don't know the answer. It's a very difficult one. And people have to be open to hearing it as well. The internet is a great place to learn stuff, but it's an echo chamber for what you want to hear. And there's so much misinformation on the internet that it becomes a big battle. I mean, it drives me crazy sometimes, the things I see people, like my relatives, posting on Facebook that are so unscientific. And there's no sense arguing about it because I don't know. I think sometimes it just makes people more resistant to listening to what you're trying to say. I don't know. It's a very difficult question, and it comes down to this trust issue. And I think in society, people are trusting less and less in science.
I think we have 3 people in line for 1, 2, and 3.
Thanks for a really interesting talks that just bring a lot of good questions to get us thinking. And before I ask my question, I just wanted to briefly say that I taught my mom to Google the stuff that she sees on WhatsApp, so you all should do that and help fight the misinformation. And my question is, I'm trying to understand how far the conversation has gone in the scientific arena and the public arena. So, like what do you think are the some examples of good progress that has been made in engaging the public in a manner that's consistent with all those values that were put up there, inclusiveness, respect, and so on. Like what initiatives have been done for that? And for the scientific community, what I was reminded of were the Asilomar conferences on that got scientists to think how to classify the safety of biomedical experiments and in relation to humans, and to come up with a more detailed list of principles that you have to satisfy, and they set up the ethics review committees and so on. Has similar stuff been going on for all this gene editing things?
I think in terms of public education, I've participated in a number of different initiatives in different countries, and my impression so far is that we don't know how to do it well, and we're not doing it very well. The one thing that I certainly see consistently as being challenging for people to understand, not because it's difficult to understand, but because people aren't clear when they speak, it's the difference between asking people if they support germline genome editing because it will allow them to save babies. And most people are kind people and they say, of course we want to help save babies. But what's happening is they're mixing up the difference between making a change in somatic cells in patients, whether they be babies, children, or adults, versus making changes in embryos, which I argue embryos are not patients, and we have no obligation to bring them into the world. And there's a very big difference between making changes in that context versus making changes to patients who have a claim upon us in the context of their suffering. And we try to respond to that as caring people in a community. And so one of the things that I have found consistently challenging in public conversations is this blurring of, do you support this kind of technology, and then lumping it all together? Because I actually believe that when I explain to people the difference, most people are, and this is borne out by the surveys, over 75% in most countries, and it gets as high as up in the 80s and 90s in some countries, believe that genome editing in somatic cells to help patients is something that we should do. What they often don't understand is that right now we could possibly, have as projects single gene disorders, and many of the things they talk about are not single gene disorders. They're very complex, multifactorial conditions. The scientists don't always put enough emphasis on gene-environment interaction, yet we know about that, and we always have these headlines. If you're thinking about misinformation, we found the gene for this, and we're going to fix it tomorrow. And I think that there's just a lot of problems around that. So every time I speak, I always spend time at the beginning distinguishing between somatic cells, germline, and as I said, I've now more recently insisted on the difference between germline and heritable, because I can imagine people being supportive of germline interventions in the lab so that you increase your basic understanding of embryology that might serve multiple kinds of goals and objectives, but you might think there's a huge difference between doing that and transferring those genetically modified embryos to a woman to have a baby. And so I think that's where I see that we need to just do a lot of work right now. Do you want to?
Unintended consequences. How much attention has been given in the community? Because we have, we look over science diseases, and there are all sorts of examples of something being done, the best of intentions, and then there's an application that could occur in a whole bunch of different reasons. So has there been much attention, if any, to all?
Well, I'll answer it quickly for humans, and then I'll let Karen to speak to some of the others, because there's some overlap and some differences. But broadly speaking, when we talk about safety and efficacy, we name the potential harms, and the potential harms fall broadly into 4 categories. So one category is what we call off-target effects. And basically what that means is you're going into the DNA with the intention of making a cut, as Karen explained, and you actually make the cut in the wrong place. It's an off-target effect, and it can have any number of consequences depending on what you've done. Alternatively, you could have what we call an on-target effect, so you've made the cut in exactly the right place, but maybe you've activated an oncogene or something, and so in that context you've actually caused a problem. In the context of germline editing, we also have the potential problem of creating a mosaic, which means that some of the cells have been edited successfully and some of them have not. And if we go back to the example of Lulu and Nana, when those embryos were transferred, it was known that one of them was a mosaic already. And we don't fully understand what happens with mosaics, but we have many people, and I can't speak to, but we have people that are perfectly fine and healthy walking around who are mosaics, right? But we don't always know what are the consequences in this kind of context with genome editing. And then the last thing is you could have genome-wide effects that were, again, unintended. So those would be sort of the 4 broad technical categories within which you could have things go wrong. And they certainly apply to the human.
So I think outside of humans, the biggest concern from the scientific community of unintended consequences is with gene drives. So these genetically modified organisms that we will release into the environment. So for example, the fruit flies. And, it's what if getting rid of that entire species of mosquito actually has effects farther up the food chain? What if those mosquitoes, can mate with some other mosquito and then the gene drive is passed on in that way? It's a similar thing with plants. Once you release, you plant them in fields, you don't have necessarily a lot of control over what happens. And, I think the example of Jurassic Park is kind of a good one when we're talking about this kind of thing, because once we release these things, if some unintended consequence were to happen, we can't put the genie back in the bottle at that point in time. And people with gene drives, people are trying to incorporate multiple layers of safety in it. one thing that people are doing now is trying to identify ways that we can turn off CRISPR-Cas9. And so there are these things called anti-CRISPRs that you can use to shut off the activity. And so the idea is, we work on them. My students are all shaking their heads, yes, back there. So we're thinking about ways that we could improve the safety of Cas9 engineering and control it if there were some kind of unintended consequence. But I think it's definitely something that we need to do a lot of thinking about.
Like a category, and there's no, so that suggests there's been some thinking, some action on it, or is it just
about unintended consequences? There definitely has been. So the U.S. Department of Defense actually had a grant call that they put out 2 or 3 years ago, and they are funding projects specifically for controlling Cas9 genome editing. And, I think part of that may be worries about bioterrorism or whatever. And I mean, I think in some ways that's, if you're going to kill someone, it's engineering a Cas9 to do something to them. You can just use chemicals or something, right? Like there's easier ways to kill somebody than to try and make some CRISPR-based thing. But it definitely is on the minds of people.
I mean, I can give you an example. In the case of GMO mosquitoes, there's a huge debate right now going on, both on the Brazilian side and on the the African side, because there's 2 types of mosquitoes: one that brings malaria and the other one that brings dengue fever. So the dengue one has been already addressed through, so there's this company, Oxitec. Actually, there's two projects in Brazil. One is the Oxitec, and another one is supported by the National Science Foundation or something like that. So it's like more state funded. And so basically they're using 2 different techniques to eliminate mosquitoes or to make them sterile, I think. Yeah. And I think that there was a very recent articles on the fact that one of the 2 techniques failed. So it totally bombed, it totally went the other direction. So,
well, one of the things that I was going to share with you is one of the mosquitoes, which is made by Oxitec, is called the friendly mosquito, and they have trademarked the name, friendly mosquito. And one of the things that's interesting is I and colleagues about 10 days ago wrote an article on the mosquito release in Brazil, and Oxitec has come after us. Wants the paper taken down. It's up on the web. So if you would put in, genetically modified mosquitoes, my name, and the word "the conversation," because that's where we've published it, and read it. I think it's a completely innocuous article, by the way. I don't think it says anything outrageous, but what's been interesting is how much effort Oxitec has put into trying to get them to take down the article, because they view it, I think, as a threat to their business plan. And part of the reason it matters is because they're trying to release these mosquitoes now in the Florida Keys. And the community is pushing back, and they don't want those mosquitoes released. So that's a very real and live, current story, the genetically modified mosquitoes.
And the public has quite a big voice.
Yeah, so just to add on to that, Kevin Esfelt is making gene drives in mice, so he wants to engineer them so they can't transmit Lyme disease. And his target for releasing these genetically modified mice is Martha's Vineyard. So it's an island full of highly educated people, and he is looking for broad consensus within that community for release of these mice. And it's his test case, and he is hoping to be able to convince people who live on Martha's Vineyard to be able to release these mice. And so actually there's a Netflix documentary coming out on Friday. I think it's called Unnatural Selection or something. So it's like a 4-part thing. It's coming out on Friday, and so it's following Kevin S. Feld and this gene drive thing on Martha's Vineyard, and Josiah Zahner, the biohacker. So it's probably, it should be entertaining, if nothing else.
I think I'm looking forward to it. I forwarded to everybody, like Elaine is making like, hey. 1 and 2.
So, I think the question I had is triggered by what Dolores was saying, about the old scientists. And it kind of comes back to your comment about oxygen. How would I put this? It's probably a very facile, cynical viewpoint, but you know, one of my favorite sayings is, how do you reason someone with a position they did not reason themselves into? Think of politically, if you look around what's going on, how easy it is to convince people with very simple arguments like DNA, and how hard it is to argue against that using nuance. So how are we going to overcome that?
I mean, I don't have a silver bullet, but one of the things that I have come to believe, and this is, I mean, I'll share with you because it's just based on my lived experience, so it's not scientific in any way, shape, or form, but I have come to believe that it takes about 30 years for something which is common knowledge within the scientific community to become common knowledge in the general public. I say that on the basis of one experiment, one subject, one experiment, which was reading Margaret Atwood's Handmaid's Tale. So I read this when it came out some 30 years ago, and I actually didn't think there was anything new or interesting in it, because yeah, that's kind of what the science says and does, and yes, you had characters, you know, playing this out, but there wasn't anything that was novel. It wasn't science fiction in any way, shape, or form to my way of thinking. And it's only now that it's become popularized. You have a TV show, you've got the next book, The Testaments, and all of that's great. But I've now come to understand that things that scientists know and do aren't part of general knowledge, and how do we get things to speed up? So how could it be maybe a 15-year cycle rather than a 30-year cycle? I mean, I'll tell you something that has nothing to do with genome editing, but how many people here are aware or know that scientists have the ability, we believe, they've proven it, they've got proof of principle in mice, that you can take somatic cells and turn them into gametes, turn them into sperm and eggs. That's not common knowledge for the general public. I think many people in the general public would be surprised to think about it, and when it becomes common knowledge, it will be because people will have written a novel about how Mary Sue liked Joe Bob, and they didn't, he wasn't interested in her, didn't want to have sex with her, but she picked up his coffee cup and took some DNA and, made a baby, and she's got his child, and then she decides to go after him for, I don't know, alimony. I mean, somebody's going to write that story, but the reality of it is that, they have taken, cells from the tail of mice, they've turned them into gametes, they've had, pups born, and those pups have reproduced naturally, and they're healthy, or it appears so. So, and that's how we do science, right? We do it in one animal model, and we slowly move to the human animal model. And I can tell you from talking to the scientists doing this, the only reason they're not advertising it is because they are genuinely worried about pushback from homophobic individuals in society. So they are holding on to their science till they hope it can have, a reception that would not be crazy and vitriolic, right? But I'm giving that as an example to say there are ways in which information takes a long time to transfer, and that's what my project right now really is about. How do we change that? How do we make information more accessible? How do we increase trust in science? How do we increase science and ethics literacy for all of us?
As autism to a wide population that's
Yeah, well, I'm talking about the an appropriate understanding of science and how it works, as opposed to.
Seems to be missing, and I fear that it's on a downslide. And the 15 years that it may take to come back in the context of genetic editing is 15 years.
I think it was Paul Field who said, so-and-so famous singer has 1 million followers on Twitter, and so-and-so famous scientist is only 50,000. I guess it's self-explanatory.
Well, I mean, I think that that's true, but it's also a challenge, right? Because scientists are struggling with what their role is around some of these conversations, right? And I think, that's hard, and some scientists are great scientists and not great communicators. Some scientists don't want to do that, even if they would be good at it. And you can't say, well, you have to be all of these things. I think there are a lot of corporate and financial interests at play, and there are Nobel Prizes to be won. I mean, there's just so many competing interests in this space. And all I have is sort of right now this naive idea that builds on the work of a philosopher by the name of Foucault, who says, if you want to understand power, you don't look to the king, you look to the masses. And because ultimately it's the masses that allow the king to stay there. I personally believe that we are in a very sad downturn time in terms of history. To me, we look like kind of, the beginning of the French Revolution, and some people saying, let them eat cake, and, they're going to bear the consequence. We're all going to bear the consequences, of that. And I think that that's kind of in the background for me in terms of understanding why it's so difficult right now to have good conversations.
There was another person who was waiting for a long time.
Yeah, so it will be sort of related to that. So it's not always the case that at the frontiers of science, those who are doing pioneering work, that very interesting ethical questions will arise. But in this particular case, I think you do understand why it happened. But in your presentation, you seem to be leading kind of ethics that's on the communicative side. Which I think I was hoping would be in the framework of Habermas in terms of communicative rationality, maybe even discourse ethics. But I had a chance to look at the book, I looked at the index, but there's no mention of Habermas. But there were mention of the flavors. So I looked through that very quickly, and it was a question about discernment. And I think you outlined that very well. But not only in a sense, is there an expectation of this type of rationality? Or is it just, as scientists, you just do what you want to do, you're not necessarily trained or socialized into this type of discourse, such that it becomes very difficult for you as an individual scientist operating in this community to sort of, cross that bridge, you mentioned this one scientist who didn't sign the moratorium, for example. Is it something unique to scientists? I don't believe it is. I mean, in any community of professionals, there would be outliers, and there's a mainstream within the community who would have these, who would operate within the constraints, or what could be agreed as constraints. Yeah. So I'm just curious, is the approach or the leaning on communicative rationality aspect of ethics the way to go, or is there anything more rigorous? I guess that's not based on consensus. I don't know what else is there. In a scientific, of course, consensus is very important.
So in my book, I avoid the F word. So while some people don't want to talk about the moratorium, I'm not explicitly using the language of feminism. And there's a lot of reasons for that, but I think if you read the book, that's what will come through. So I'm worried about oppression. I'm worried about the distribution of power, and that's ultimately where my understanding of feminism takes me, right? I'm looking at who's being harmed, who's being oppressed, whose voices are not heard, who's controlling the agenda. And really, if it were to be the case, I think that's the thread that you would see. I'm not interested in this book to, I try really hard in this book, with a few exceptions, not to offend, not to be vitriolic, not to expose at great length my own ideas. I think if you read it carefully, you can see what I stand for and what I believe in, but it really is an effort to sort of say, these are interesting questions, and these are the things that some people are saying about these questions. And your point about, is this unique to the scientific community? Of course not. I mean, we have, they're humans like everybody else. You're humans like everybody else. I mean, they have views. But what I think is particularly interesting is the language that's used to describe some scientists, the polarization that has happened around some issues. And I'll just give you two examples. In, I believe, around 2005, I had a little, public debate with John Pollani in the Globe and Mail, because I was saying, and I still believe, that when you do science, it should have some social value. And, Polanyi's basically saying, are you crazy? Or that's the polite way he was sort of saying that. But he's basically saying, no, that's not what science is about. Science is about serendipity. Science is about excellence. And when you try to manage science, you manage excellence right out of science. And your ideas are all wrong and flawed. Science is not about social value. It's about excellence. I don't share those views. I think science should be about excellence, but I think it should have social value. The flip side, if I were to take another example, is Jean-Q Hay has been described and discredited as a rogue scientist. And I don't discuss that at length in the book, but I think that's absolutely wrong. He is not a rogue scientist. He did exactly what was expected of him, exactly what he is trained to do. He was trained in the United States. He understands that you give prizes for being first. He understands that only one person is ever going to be first. He read the biography of people like Steptoe and Edwards. He truly thought he was positioning himself for a Nobel Prize. The idea that this really happened in secret and that nobody knew about it, not possible, right? I mean, the kind of effort that has gone into marginalizing him and having him bear the weight of everything that went wrong. Don't get me wrong, he does bear responsibility for the choices that he made. But when this news broke, I was just reading the paper, and then I immediately went online to try and see it. And when I saw a picture of him, I thought, oh my gosh, I know this guy. And then I went back through my files to say, to confirm, do I really know this man? And sure enough, I had been at one of those two small meetings that he describes where he says he consulted with Americans about it. He sat 2 seats away from me. And if you had asked me anything about this man, I would have said to you, he's a naive pup. He doesn't understand the complexity, the social issues with what he's talking about. And at that meeting, he wasn't talking about making babies. He was just sharing the information he was doing around manipulating embryos in a lab. There was nothing he was doing in and of itself. But in that context, if anything, there was huge naivete. And I think people were happy to have him be the one to walk over the line. Because now, what do you have right after the 2018 summit? I mean, that's why I start almost all my talks by saying, this happens in 2015, we get this response. 3 years later, this happens, we get a completely different response. And it's because at the end of the day, somebody has to go first. And if we can hang them out to dry, that's great, because afterwards, everybody can always say, I will do it better than him. So, I mean, that's a bit of a convoluted answer, but it's to say there are a lot of players and different perspectives.
Trying to think if I have anything to add. I mean, one comment I will make is, I trained biomedical students, and I was trained as one myself. I never took an ethics course. My students don't take an ethics course, although I think the university has recently instituted a short ethics course, but it's about not manipulating your scientific data. It's not thinking about the broader societal impacts of what your research may be, or what the implications are. And I think there's a quote from Jurassic Park that I keep bringing up Jurassic Park, but we were so busy trying to see if we could do it, we didn't think about whether we should do it, right? And I think scientists can become mired in this, I want to see if I can do it, and then you've done it, and you may have opened a can of worms.
But I get more and more students from the science. I think technology and SPS. There's a lot of ethics in that. I really want to be a chocolate.
Yeah.
I'm Susie, because I'm teaching a course in biotech in But I get more and more students. I noticed that there's more and more students.
Yeah. I mean, I think it's good, but it's not an instituted part of our training of PhDs.
That choice. Right? Like it's not like something that you like.
And it's not just here; it's everywhere.
Do you remember anything to add, or like? Because now we have another person in the back. Okay, so.
Yeah. I'm just curious about the babies that were born HIV resistant in China. They're twins, obviously, are they not? Okay. How old are these babies, and where are they? Do you know?
The babies were born in October of 2018. The news was made public at the end of November 2018. At the time, Jean Cuhay said that there was another pregnancy, and more than 9 months have passed, and we do not know what has happened to that other pregnancy. And we also do not know what has happened to those girls. And my expectation is that we will probably never know. And the reason for that is, on the one hand, strong ethical arguments are being made that, they should not be lab rats for the rest of their life, that the Chinese government and society may have some interest in doing long-term follow-up, but they don't have any interest in attracting attention to this issue right now. And so my expectation is that we will probably not know until somebody decides to issue a press release 5 years down the road to say they're doing well or something like that. So yes, we don't know. What we do know is that there was supposed to have been another pregnancy. We've not heard any news. Did that woman choose to terminate the pregnancy? What did or didn't happen? Did that woman give birth? Is there another child out there? We just don't know.
This will be the last question.
This is kind of a long question. The question is looking at the Margaret scale globally. So there is already, as you know, there is a huge gap between the first world and the third world economically, and in terms of human life, quality of human life index. So assuming in the near future or part of the future, the gene editing and gene therapy, it is given a green light for scientists to do in different ways. There is going to be, I think there is going to be, a gap between the first world and the third world, and considering, they are behind scientifically and economically and everything. So I think that will actually be, in a way, it's kind of radical, but in a way it will be actual application of eugenics. And so in your own research, do you think, is there any parameters of preventive measures in place right now to prevent it?
So certainly, I'll sort of, by answering the question, indirectly repeat it. So some of the question has to do with the increasing gap between the haves and the have-nots, between high-income countries versus low- and middle-income countries, what attention's being paid to that, or in anticipation what might happen. I think many people are deeply concerned about the issue of access to these technologies, assuming that they would go forward in some way, shape, or form. These technologies are expected to be extremely expensive. If I were to take just examples in the realm of gene therapies, so looking to offer somatic interventions to patients, one of the early successful gene therapies is actually Canadian developed it, Dr. Hayden out in BC, and it's the drug, if you will, the biologic is called Glybera, and it's safe, it's effective, and 31 people in the world have had it, and it's no longer on the market. And it's no longer on the market because it was not a viable business. It's $1 million. You just don't have a lot of patients with a spare $1 million. And you could argue that, you would hope that the healthcare system would pay for it. Maybe if you have Cadillac insurance in a place like the United States, maybe. But a lot of people in the United States don't have Cadillac insurance, they don't even have insurance. And in a country like Canada, I mean, even if you have a government-funded healthcare system, there's only so much money in the pot, and decisions get made all the time in terms of what can and cannot be paid for. So one of the things that I encourage people to be thinking about in this area, just to show how complicated it is what do you do as you're busy developing technologies that you anticipate, because of the current capitalist structure we have, will not be available by and large. They'll still be really cool science, but will they in fact turn into treatments or therapies that we could offer to people? And in that context, I am still asking scientists to tell me whether they are looking at this science through the lens of public health. So we are looking to develop some kinds of prophylactic interventions for all humans, or is this a form of personalized medicine? And I think that's a really, really key question. And I don't think we're struggling with that question enough. I think if we did struggle with that question, we'd be getting at things like social value. What's the social value of this science? And, you know, assuming it's good science and we can get to safety and efficacy, where do we want to direct it? Where do we want to put the time, talent, and treasure it will take to develop these technologies to build a better world? And right now, I don't think we're spending enough time thinking about that. In the book, I have a whole chapter on slow science, and a lot of the scientific community is really upset with that chapter. They don't want to do slow science. They say, Françoise, people are dying. We need to do this now. And I'm saying, well, you might be killing people, right? I mean, you know, and I find it fascinating, this resistance to the idea of slow science, because slow science is about, you know, taking time to think about the goals and objectives and what you're investing in and where you're taking your science. So it's a bit of a long-winded answer, but it is to say you are naming a very real problem. Access will be a huge problem, and we should be thinking about that as we make choices about where to invest.
Okay, the very, very last one, a quick one.
Quick question. So do you think that the advent of CRISPR technologies drawing attention to our lack of understanding of basic science in other fields of study? So take, for example, like the mosquito basic ecology. Let's take, for example, human genetics, understanding of complex diseases. So is the problem with the CRISPR research and the CRISPR technology specifically, or is it just that we don't understand our science well enough as a whole?
But if you read Françoise' book, you will understand everything, because it's very easy, really, like it's the first chapter.
I'd like to point out she actually works on CRISPR. I don't know. I mean, a lot of the issues that we're going to be struggling with, or we are struggling with in CRISPR, should we do it? You know, who gets access, are all things that we're already struggling with in society. I mean, you know, when we talk about editing embryos, in many cases, like Françoise said, there's not really a reason to edit embryos. We could do pre-implantation screening and not implant embryos that have that genetic defect. And so we have mechanisms to deal with a lot of these issues now. And, you know, CRISPR, we've been engineering organisms for years. Genetically modified plants have been going on for 20 years. CRISPR allows us to do all of this work a lot faster. That things that used to take us 2 years, we can now do in a few weeks. And so I think the framework for looking at the ethics of dealing with a lot of the issues that are coming up is no different than other medical treatments and issues that have come before.
I think for me, it's that last point that I'm trying to push on, and that's where I'm meeting a lot of resistance, which is that a lot of people, in fact, do say, and at some level they're not wrong, look, you know, this is just a quicker, cheaper, faster way of doing what we're already doing. We know how to do this. We've got research ethics committees that look at it. We know how to fund it, et cetera. And my view at the end of the day is that if we do CRISPR right, we'll actually change the way we do science, because ultimately it's about the way we do science. And you heard me make some of those comments. I don't think we should be doing science in a way that's competitive. People disagree with me. I think if the goal was knowledge production, if we could find ways of being collaborative, we would actually do better on our knowledge production. People say that I'm wrong, that, how did we get to the moon? It's because we were competing with the Russians to get to the moon. I kind of think we could have gotten to the moon if we wanted to collaborate with them as well. But I think we haven't talked about it. But there's a lot of nationalism involved in science. There's a lot of competition involved in science. There's a lot of secrecy involved. There's just so many ways in which we do science generally that I think is just exacerbated by this particular science, partly because it is speeding up everything. You saw, with the references to the papers, you saw what's happening. And that's why when I want to talk about slow science, people are getting exercised because they're afraid of being left behind, or they're afraid of not being first, or they're afraid that they won't get the patent, or they won't get the money, or they won't get the deal with industry. And all of those pressures, are just adding up. And I will, continue to say if we really try to do CRISPR well, we could improve the way we do science generally. And I just really believe that. Anyhow, thank you everybody for coming. It was wonderful. Appreciate it.